Hi everyone, in this video we'll be talking about the process of DNA packaging. DNA, or deoxyribonucleic acid, is the hereditary material found in most organisms. DNA is found in almost every single cell. If you were to stretch the DNA in each cell all the way out, it would be roughly 2 meters. Cells are obviously way smaller than 2 meters, so how does DNA fit within the cell? The answer is DNA packaging. Through a series of interactions with proteins, DNA becomes condensed into a form known as chromatin. This structure allows DNA to fit within the nucleus of a cell. So how does this process of DNA packaging actually occur? First, you have DNA floating around the nucleus. DNA is a negatively charged molecule. Also within the nucleus are positively charged proteins called histones. The histones arrange themselves into 8 protein complexes known as a histone octamer. The negatively charged DNA then interacts with and wraps around the positively charged histone octamer, forming a structure known as a nucleosome. DNA continuously wraps around the nucleosomes, forming a structure often likened to beads on a string. At this point, the DNA is shortened about 7-fold. This means that if a piece of DNA was 1 meter long to start, it would now be 14 centimeters long. This is much better, but given the average cell is about 20 micrometers in diameter, more still needs to be done. The nucleosomes then, which each contain about 146 base pairs of DNA, coil into chromatin fibers consisting of the packed nucleosomes, condensing the DNA into a shorter, thicker fiber known as the 30 nanometer fiber, as it is roughly 30 nanometers in diameter. The 30 nanometer fiber loops around on themselves, are compressed, and then coiled further to form the 250 nanometer fiber, which produces a chromosome, as shown here. The degree to which DNA is condensed varies throughout the cell cycle and plays a functional role in different processes. For example, DNA is in its most compact form during metaphase of mitosis, which is essential for proper alignment of chromosomes along the metaphase plate. The process of DNA packaging in prokaryotes is much simpler than their eukaryotic counterparts. Prokaryotes lack membrane-bound organelles and do not have histones to help package the DNA. Instead, the DNA undergoes a process known as supercoiling, where the DNA is overwound, resulting in a structure that takes up less space. So, in review, linear DNA must be packaged in order to fit into the nucleus of a cell. This is accomplished by a series of steps, including DNA wrapping around histone octamers, nucleosomes coiling in order to form a structure known as chromatin, chromatin then condensing further into 30 nanometer fibers, which then loop around on themselves, are compressed, and then coiled further to form the 250 nanometer fiber, which produces a chromosome. In prokaryotes, the process of DNA packaging involves DNA supercoiling to ensure the DNA fits in the cell. That's all. Thanks for watching.